Hi there. In this video clip we're going to continue our discussion of central metabolism, and we left off with acetyl-CoA being produced from either sugar or fatty acids, being present in the inner mitochondrial matrix and being just about ready to integrate into the citric acid cycle. And here is the molecule, the 2-carbon molecule of acetyl-CoA being condensed with a 4-carbon molecule, oxaloacetate, forming a 6-carbon molecule of citric acid or citrate. And that's why this is called the citric acid cycle. It's also called, as I mentioned, the TCA cycle for tricarboxylic acid cycle, or the Krebs cycle. Once again, we will not be memorizing structures for this course, but if you're taking the MCATs, you will need to memorize them, which includes the molecules not even written out here, but alpha-ketoglutarate, succinate, fumarate, malate, and then finally getting converted into oxaloacetate. There are 2 steps where we lose carbon as carbon dioxide, and 5 energy generating steps when we produce either NADH or FADH2 or GTP, which, although not as common as ATP, is still used very often in energy requiring and regulatory processes. Now, NADH and FADH2 both function as electron carriers. NADH has higher energy electrons. There's not enough energy being produced between steps 5 and 6 to produce another molecule of NADH, and rather than throwing that energy away, cells are able to save that energy in the form of a less energetic molecule, FADH2, although FADH2 will not make as much ATP as NADH will. Now, like acetyl-CoA, you will not need to memorize either of these 2 structures, NAD or FADH2, but I would like to show it to you. This is the N part, the nicotinamide group. The adenine diphosphate group is again similar to what you saw for acetyl-CoA, or it's similar to ATP. And when you carry electrons, you're going to add a hydride ion here at this carbon. And the fact that you've added an extra electron in the form of hydride ion rather than hydrogen atom is manifested in the fact that the positively charged nitrogen here is now neutral and will stay so until hydride ion is again lost when we do oxidative phosphorylation. You don't need to remember these structures. Here's FADH2. In this case, we still have the ADP basic unit, and then a flavin ring. That's a flavin ring. This is the F in the acronym, and we're going to add, in this case, 2 hydrogen across this unit and 2 electrons. So this is the reduced form, and this is the oxidized form. This is the more energetic form, which can then donate electrons into the oxidative phosphorylation chain. So here we have an overview of what we've covered today. Glycolysis out in the cytosol, producing pyruvate from glucose molecules, being transported into the inner mitochondrial matrix, where acetyl-CoA, pyruvate is converted to acetyl-CoA. It's co-opted into the citric acid cycle, in which the 2 carbon molecules present in acetyl-CoA will be released as 2 molecules of carbon dioxide. In the meantime, as we're doing that, we're making molecules of NADH and FADH2. NADH will introduce its electrons, its higher energy electrons, at the beginning of the electron transport chain. FADH, which is less energetic, will introduce its electrons further along the way. We'll get 3 molecules of ATP for each NADH, and approximately 2 ATP molecules for each molecule of FADH2, And these electrons that are going to be present in the electron transport chain are finally going to end up on the molecule oxygen, producing water. This being again, the reason that we need to breathe. This is why we die when we don't have oxygen, is because the electrons get backed up in this chain and we're not able to make sufficient ATP to meet basic metabolic needs. These are the enzymes involved in this step. Cytochrome C oxidase is the protein to which cyanide binds, and that's why cyanide is a poison. So essentially, oxygen is a molecule we need as a place to dump electrons when we're done with them, when they're not very energetic. So in the last two slides, I want to go back to the process of anaerobic respiration, which does not involve the citric acid cycle or oxidative phosphorylation, because you don't have oxygen present to accept the electrons. And as you recall when we were talking about glycolysis, is NAD plus is a required molecule to put in as a reactant for the process to go from glucose to pyruvate. And if you don't have oxygen present, the only way you're going to get ATP is if you continue to break it down. So that means you need a constant supply of NAD+. The problem is, every time you break down a molecule of glucose, you use up your supply of NAD+, and you're making a lot of NADH, which you have in vast excess, and which is not that useful if there's not oxygen around. So what's going to happen in fermentation is you're going to take this pyruvate that you've made, you're going to take the electrons from NADH, and you're going to produce lactic acid. And when this builds up in the muscles, that's when you get cramps. But since we've gotten rid of the electrons from NADH, we've regenerated the NAD+, which in the absence of the oxygen is the valuable molecule of these two. Now later on, after the run is over and you have plenty of oxygen, the electrons that you've dumped into lactic acid can be put back on NAD+, reforming NADH so you haven't lost it, and then NADH can go ahead and produce lots of ATP using oxidative phosphorylation. So in that sense, lactic acid becomes a temporary electron parking spot. And bacteria have evolved a different strategy for the same problem of regenerating NAD+, for which we're all probably pretty grateful. Pyruvate gets converted to acetaldehyde, and then it accepts electrons from NADH, producing ethanol, and this is the process which produces beers, wines, and other alcoholic beverages. But the important point and the ethanol is excreted because in high concentrations ethanol is toxic to these organisms. But the good thing from their point of view is that you've regenerated NAD+, and you can continue to allowing you to continue to break glucose down to pyruvate, producing the only ATP you're going to make in the absence of oxygen. Okay, thanks for